And then,
Oh my back. Oh my God.
no, it's taking its time, right? Is there anybody in the waiting room? So we can like know, and then
I have
that's
no
muted. Okay,
clue.
great. Let's see. I think we could probably start, yeah, record. And then, yeah, we're essentially gonna start off the webinar.
Okay, cool.
Yes, welcome everyone in as they come in. Do we have any more participants?
Yeah.
Are they in? They're in. What is it?
Yeah.
All right, welcome. If you just joined us, welcome. We're going to be having our last presentation, last webinar of the year, and of today as well. So yes, we're very excited to have an EHS alumni tell us all about, was it, you actually, we'll hand it off to you. So Hadley Copeland, and yes, thank you so much for joining us.
Hello, thank you so much for having me. I'm super excited to be back here. As they mentioned, I am an EHS alumni and I was a part of NBTI when I was in college about 10 years ago. And I had a wonderful experience being able to share with the world the different biodiversity talks that were available. And it's awesome to be able to come back and kind of share my learned expertise as well. I'll be talking about salamanders today. I did some salamander work when I was in undergrad, and I'm doing them now in my master's program. So I will be kind of giving you guys an introduction into how salamanders function, as well as kind of what that means for the world and how I fit into that. So I'm going to go ahead and share my screen really quick. All right. All right, so there we are. Once again, I am an EHS alumni. I did get my bachelor's at Coe College, and I majored in biology, chemistry, and biochemistry. When I first started off there, I was pre-vet, and then I did research and found out how much I loved that and switched gears, did some tech jobs for a while, working with the DNR in the State Hygienic Lab, doing water quality assessments. And then I moved forward to the University of Iowa, working at the veterinary diagnostic lab there. And now I'm at the University of Georgia, working at the veterinary diagnostic lab here. I am also currently getting my master's in wildlife sciences. I'm concurrently working as a histology technician, which I'll go into a little bit at the end. But a brief overview of histology is it's the process of making microscope slides. So we'll get in necropsy, animal tissues, or biopsies, and be able to make microscope slides from them, stain them different colors so that the pathologists have the opportunity to make diagnostic results from them and give your pet the appropriate treatment based off of what we know about their diagnostics. So my background a little bit, and the reason why I have the ability to talk to you guys about salamanders today is because all of my background in research is based with salamanders. So for my undergraduate thesis, I worked with blue spotted salamanders, which is Ambystoma laterale. They were in Iowa, where there's two populations, though there are a lot more populations throughout the Midwest and up into Canada. They're the little guys on the left hand of the screen with the blue spots. I worked doing breeding and habitat quality with them to kind of assess the quality of the ponds that we had some long-term abundance data for, but no true water quality data for. The ponds there were surrounded by agricultural land, so it was good to get kind of a baseline on the nitrogen impacts from being near agricultural land, as well as baseline levels of detritus and if the impacts of pH that decay had on certain ponds in the area, and kind of look at the movement ecology of those breeding adults. For my current thesis, I am working with stream salamanders, which are those two other individuals at the bottom. The one in the middle is a black-bellied salamander, and the one on the other side is a two-lined salamander. Those guys both are found in streams in the southeast United States, and I am looking at how they are responding to imidacloprid exposure, where imidacloprid is a pesticide that is applied both for agricultural use, but also in forest treatment plans. There is some impacts of beetle that is killing off some of the trees in the area that is super, super critical for a lot of habitat for animals throughout Appalachia. And I am helping study how this pesticide that's saving those trees could have some non-target effects on these little guys. So now to get into salamanders. The name salamander comes from the Greek origin salamandra, which means fire lizard. When they first named these individuals, they would see them crawling out of the logs that they put on fires, and they assumed that they lived in fire or had the capacity to live in fire. They do not. So salamanders are not able to survive in fire, which is why they were fleeing from those logs as they started to burn around them. However, they do love to take up residence in decayed logs. They often will hibernate within tree, like they'll follow tree roots down into the ground to help hibernate and stay at a more consistent temperature over the winter. And they will generally be around fallen trees and logs when they're in their terrestrial forms. They are from the kingdom Animalia, of course. They're also in the same phylum as we are, Chordata, which means they have this dorsal nerve cord that we have, just like our spine. And then they have some other characteristics that align with us throughout our embryogenesis. The class is amphibia, so that consists of frogs and salamanders. The characteristics of amphibia are they have to be cold-blooded, so they're ectothermic. They stay in warm areas to be able to regulate their temperatures and be able to do things like digest and move and reproduce. In times when it is too cold for them, they will not be able to do those things. So you'll find lots of amphibians will try to behavioral thermoregulate by either going into warmer areas, going underground when it gets cold, and some amphibians, such as some frogs in the very far north, actually have additives into their blood that allow them to kind of resist the process of freezing so that they are able to survive over winter as well. Amphibians also do not have scales, which is unlike our reptile friends that are also ectothermic. Because they have the ability to breathe through their skin, they also have to have moistening glands on their skin so that they can be able to respirate. And then they have a three-chambered heart also, which kind of goes along with that skin respiration thing. They are part of the order Caudata, and also technically the order Urodela. Caudata consists of all of the salamanders, both extinct and extant, or currently living. Both Urodela and Caudata, that order, they have to have a developed tail. So unlike frogs, they have that longer tail. Their legs are all similar in length, unlike frogs. They have gills in aquatic larvae, so they have those fluffy external gills that come out around their neck. They have teeth on both jaws, again, unlike frogs, and they do not have tympanic ears. So if you look at the side of a frog's head, it has this nice round drum. We also have an eardrum set inside of our own ear. These guys do not have a tympanic ear. They do still have an ear bone, and I'll show you guys some pictures of those later. And they also have ribs, which some amphibians do not. So to go a little bit closer into their taxonomy, we have our suborders and our families. There are 3 suborders. The Cryptobranchidae are kind of our ancient order of salamanders. That includes our giant salamanders, such as hellbenders and some Asiatic salamanders. There's also the sirens, the Sirenidae. These guys do not have legs. They often live in larger water bodies, and they get quite large. And then we have all the rest of the salamanders that we love and know, such as our mole salamanders, like the tiger salamander, that is a state amphibian for many states around the United States. We have our amphiumas, which do not have back legs. We have our lungless salamanders, like the species I'm working with now, mud puppies that stay in their aquatic form their entire life, similar to axolotls. And then we have our torrent salamanders and our newts and true salamanders. The anatomy of salamanders is kind of interesting. So we'll go over some of the key characteristics that make them special to this group of amphibians. Starting with their circulatory system. First things first, they have that three-chambered heart. So they have that blood mixing situation going on because they're able to respirate from areas that mammals will not have to do. So if you look at the picture in front of you, you can see the pig on the right, the mammal, has a very clear coloration between its two blood types. The red is oxygenated blood going down into the body, and the blue is venous blood coming back from the body. You need oxygen to be able to expend energy and to be able to utilize your body and digest and move. So having oxygenated blood is super, super important. In mammals, it's very isolated, where we have the right side of our heart collects all the venous blood. It's shunted out to our lungs where it becomes oxygenated and then goes back into the to the left side of our heart where it is then distributed through the rest of our body. In salamanders, that is different, and in frogs as well, because they have the ability to breathe through their skin. Because they can breathe through their skin, they have no true, fully, like oxygenated blood because their skin allows the blood to exchange directly. So they always have consistently non-oxygenated blood and oxygenated blood flowing through their body. So you'll kind of see that in the picture in front of you, notated by that purpley color, where that's mixed blood, where it could be oxygenated or non-oxygenated. It's a little bit of both. How that kind of works is the atria will either put the blood out into the lung capillaries or put it out into the body, and then the body's capillaries are doing that direct exchange. For larval salamanders, they're able to have additional capillary exchange through their external gills. On the left side, you have an axolotl where they have these gigantic fluffy gills. Lots of larval salamanders look like this as well, where they have these gigantic fluffy gills. You can see they're bright red. They have lots of capillary action. The red is defined by that blood going through it, which is able to do that oxygen exchange through the skin. They are also able to exchange oxygen through the layers of skin in their mouth as well, so they do sometimes breathe in air through their mouth. The little guy on the right is actually one of the species I'm working with, a Desmognathus species. That's that black-bellied salamander that was in the central photo of my background slide. They have a weird set of gills where they're a lot smaller, they're not as fluffy. They still offer extra surface area to provide some additional surface area for gas exchange, but they do not provide it in the same way that other salamanders do, as noted by the guy on the right side, or the fluffier gilled guy. That is because Desmognathus is an interesting genus where they were initially direct descent, where they were in their embryonic form, did not go into this larval stage that had the gills initially. They eventually evolved to have that form just because it was better for our current evolutionary status with how they develop in the streams. But when they were first developed, they had eyelids as embryos. They didn't have gills at all, and they'd come out and just be a miniature version of the fully formed adult. Now they do have those little bit of gills, and they do serve a function, just not as much as some of the other ones. So through the skin, you can see, again, directly through the layers of the epidermis, the oxygen will go directly into the veins and into the capillaries in both the gills and the skin. And there is an interesting genus called the Plethodontidae that do not have lungs at all. So they have a little bit more interesting of a gas exchange process where they only have access to their skin for gas exchange. Their blood does not pump anything into lungs because lungs do not exist. So these guys are endemic to fast-moving streams. They rely on highly oxygenated water. So they tend to be in areas that have riffles or small rocks that cause the water to be more aerated and allow them to have that extra gas exchange through there. They also stay within stream areas their entire lives, as opposed to some of the species that become more terrestrial as they get older, such as our mole salamanders and our newts, that are lunged and have the capacity to be less moist and have other opportunities to take up air into their lungs. All right. Here we're talking a little bit about their ears. So different species have different ear bones, but none of them have that tympanic membrane, that eardrum that we see on amphibians and most other species of mammals and birds. These guys just have next to their jaw, you'll see they have a very smooth side of their head, and then right behind their jaw, they have their ear is fully internal. It lies right behind their orbit for their eye socket and is able to pick up auditory information through there. So they end up relying a lot more on their visual sense when they're hunting, as opposed to lots of other reptiles that rely more on auditory senses. It also kind of goes to show a little bit about how their reproductive interests are different than other amphibians, such as frogs, who depend on calling and have large tympanic eardrums to be able to take in that auditory information and have the capacity to choose mates based off of that. Salamanders depend on different things when choosing mates, and we'll go into that in just a second. But they do not care about auditory information, and they do care a little bit more about color. They have a similar eye structure to us, in which they do have 3 color cones, so they're able to see colors a little bit more vibrantly than a lot of species, and they are able to kind of choose their mates based off of that. The other thing that they depend upon is movement to be able to choose their mate. So we'll go into a little bit on how these animals can move in different ways. They do not have claws. They do not have the ability to climb very readily, but they do often have either webbed toes or flattened tails that help them swim through the water. In the example in the middle, you can see that flattened tail is that of a newt. When they are in their aquatic form, they will have that big flat tail. Larval salamanders also always have that flat tail, and it helps them swim kind of like a paddle to be able to move faster. When they take on a fully terrestrial form, you'll find most of the adults do not maintain that tail shape because it is not beneficial to them anymore. And there's other mechanisms that they utilize their tail for that would be a hindrance if it were to remain in that flattened shape on land. Mole salamanders, such as the marbled salamander that is in that right picture, are very good diggers, hence the name mole. So the Investimid genus needs to have that smooth tail to be able to dig, and they actually do move soil around a little bit with that tail as well. A lot of salamanders also utilize their tail as a defense mechanism. They will drop their tail when threatened and can also secrete a latexy secretion that's kind of milky in color from them that will kind of hinder eating from other species. So, for example, if a snake were to try and attack a salamander, he could raise its tail up and squirt this liquid from it, and the snake's mouth could become difficult to open, allowing the salamander a chance to get away. Other defense mechanisms these guys utilize is mainly their camouflage, so they all have very different colorations. They tend to move around mostly at night, and they tend to dig and stay underneath a leaf litter when they're moving around to be able to stay out of sight, out of mind, and away from the predators. So salamanders reproduce and go through their life cycle in very interesting ways. There are multiple types of reproduction in the different species of salamanders, and there is a group of salamanders that also goes through some type of unisexual reproduction that we'll discuss further in a moment. The most common type of fertilization of reproduction within this group is internal fertilization. It is different than mammalian fertilization, where the male injects sperm into the female. Instead, the male lays spermatophores, which are in the picture below, which is like a group of sperm encased in a gel that the female then will pick up into her cloaca and store in a sperm pocket. They do a sort of dance, so they utilize their hands for gripping, they utilize their tails for movement, and they also utilize, if they have them, this guy, the bright orange one, you can see he has these little barbs at the bottom of his chin. That is another thing that they'll use to help entice the female, and then they will guide her to where those spermatophores are so that she can pick them up if she so deems. There are often cases in which the female deems she does not want to pick up that male spermatophore, and then they lay on a leaf like that and chill for forever. Females in their sperm pocket are actually able to store that sperm for later use. They can store it for years, up to 2 years, which helps them be able to determine if it is a good breeding season, if their young will be able to adapt well to the situation that they've been put upon. Is the water high enough for them to be able to survive? Are they breeding early enough for them to have enough food? So there's lots of things that go into the consideration of breeding that females will utilize to kind of determine if they want to lay eggs that year. These guys are different from frogs in that they do not lay thousands of eggs all at once. They lay eggs over several days, and it takes a lot of time for them and a lot of investment for them. So they're more choosy in how they breed, typically. Some species will lay several eggs, and they'll wrap them in leaves or on grass strands and kind of leave the eggs to fend for themselves. Some species, like a lot of those lungless Plethodontidae, will actually have the females guard their young, and they'll like sit over the eggs and sit over the embryos and the young larvae, and will guard them, protect them, keep them safe in a little rock cave that she has chosen. So there's a lot more maternal investment and choice in these amphibians in comparison to frogs that are able to lay as many eggs as they want, and if only 10% of them survive, there's still 100 frogs out there. These guys only lay, depending on the species, between 5 and 20 eggs, so they have to be able to be more choosy and more specific about when they're laying eggs and where they're laying them. The other type of reproduction is external fertilization, which this is only done by those other two clades that are not the typical salamander. So the sirens, such as this picture below, the legless guy with the big fluffy gills, they're an aquatic variety their entire lives. They stay within the river systems that they're in. So they breed more similarly to fish, in which the male releases sperm and then the female releases eggs. And when they come into contact, then they become fertilized, and then the eggs will eventually develop into embryos and into young. These guys are able to kind of breed a little bit more like frogs, where they're able to release a lot more eggs. And because of that, it's because they have to, because they are not able to do that direct fertilization. See, the weird guys, so the blue spotted salamanders that I worked with were a part of this group called the Jeffersonian salamanders. They're an abystamid, so they're a mole salamander that likes to dig, and they live in the kind of Midwest, north, like southern Canada area. In there, they're able to do a couple of different types of reproduction without males. So the first type is called parthenogenesis, in which the female decides to just replicate herself. There's no genetic material added. She just decides to fertilize her own eggs of her own ambition and will put out direct replicates of herself, or clones almost. There's also this type of reproduction called kleptogenesis that they're able to utilize. So these species that are like all female species, there's no males at all within the species. In order to reproduce with genetic variation, to keep a healthier population, they will choose to steal spermatophores from males of other species so that they can either incorporate that genetic material, or they'll just use that to start the process of replication and won't incorporate any genetic material. They can sometimes uptake genetic material from multiple spermatophores and create hybrids in which they have multiple chromosomes from different individuals. There's 5 other species up in that area, including the tiger salamanders that you can kind of see in the middle of this group of 5 salamanders on the side. They are able to create individuals of hybrids utilizing multiple genes from multiple individuals, which is incredible, and will allow us to potentially create breeding programs for those that are in areas that are slightly more threatened, such as those Iowan species I worked with, in which there were only 2 populations within the state. So being able to have this process where they can steal genes and create hybrids is really, really interesting and is kind of novel. There's been research into it, but the process isn't fully understood at this point. So being able to understand it more fully will allow us to improve our breeding operations, to be able to head start these individuals and make sure their populations remain healthy. It also brings up an interesting question on hybridization versus speciation. Do these guys count as that unisexual individual anymore? Are they part of a hybrid in which they are multiple species at once? What actually counts as a species within this genus? So there's some interesting questions within this group that is brought up through these individuals that can do kleptogenesis. So if it's something that interests you, I highly recommend looking into the Jeffersonian group that can do the kleptogenesis, because we need more people researching them and trying to understand how incorporating multiple chromosomes is able to provide a healthier population for those guys. The life cycle of these guys can vary. So there's some individuals that stay in their larval stage for their entire lives, such as those amphiomas, those sirens, those axolotls, where they never quite get past that stage with those fluffy gills. They always stay in that area, and they become full adults with gonads in which they're able to reproduce, still having those fluffy gills and being fully aquatic. the ones that go past that stage become terrestrial juveniles and terrestrial adults, and eventually will return to the streams or the water bodies and lay their eggs. Some individuals stay near the streams, such as those lungless salamanders that need moisture to be able to survive. Some of them stay in moister areas, such as crevices in granite or caves, such as the emerald salamander. And some of them actually revert into a more aquatic form once they return to the water, where they regain that flattened tail to be able to gain better motility, such as newts. There are some newts that also have a bit stranger of a process of metamorphosis, such as the striped newt. They lay eggs, will go into that larval stage that is consistent for all salamanders. And then they have a couple of options. They can either go into this F stage like most newts go through, where they're a fully terrestrial juvenile, they do not have reproductive organs, they are not able to reproduce, and they kind of just traipse around in the woods, hanging out, getting food, before they decide to become a terrestrial adult, in which they will develop their gonads, and then they'll go back and forth between their aquatic adult, where they have that fluffier tail and they're able to reproduce in the water, and this terrestrial adult form, where they still retain their gonads, but they're traipsing around in the woods, and they do not reproduce because their eggs would not survive there. They also have this intermediary form, which, depending on the location that you're pulling these newts from, is more typical or less typical for a population. One of the populations that one of my peers works with is in an area that had heavy agricultural input from around the area, so it was not desirable for them to go into F and terrestrial adult form, and instead the population takes up this normally paedotypic form within their intermediary stage, where they still have these fluffy gills, they still have larval characteristics such as that long tail, they don't have eyelids yet, but they have gonads, and they are able to reproduce. And that's more desirable because they do not have to enter these agricultural fields or these urban areas where they do not have the capacity to collect food as a terrestrial adult in a forested region would. So instead, they go into this paedomorphic form where they're not quite an adult, but they have gonads, so they're kind of an adult, and they're able to breed, and they're able to stay in the water and be safer from external outputs, such as agricultural inputs to the environment. So depending on where you're finding your newts, you might have these different forms that you're dealing with, but generally all salamanders will start off as eggs and develop into larvae, and then is where things get a little bit weird. So the last thing I kind of want to talk about that's somewhat unique to salamanders is regeneration. There are other species that can regenerate, but they're all technically invertebrates. So you might have seen cases in which octopi can grow back their arms, or starfish can grow back their legs, but all of those are invertebrates, and they do not grow limbs in the same way that vertebrate animals do or chordates do. So these guys are the only chordates that are able to grow limbs and grow organs the same way that us humans do and other mammals do. So they're a great indicator for us to be able to do research on regeneration within people and within other animals. How it works within salamanders is when they lose a limb, instead of their wound causing scarring, there becomes a wound bud called a blastema, and it, instead of calling cells to it to create scar tissue and to heal over the wound and to strengthen it, it starts bringing these blastema cells and dedifferentiated cells back to this area, in which they're able to reform what they want. So when we're very young, when we're embryos, we have these dedifferentiated cells and stem cells that are able to do that same thing. Most embryos have the ability to do so. So if you were still in your mother's womb and you lost a finger, you could grow your finger back because you're an embryo and you still have all these stem cells that aren't yet a fully developed cell. It hasn't decided what it wants to be yet. So it can go off and be whatever it wants to be and whatever it needs to be. So if you are in the process of growing an arm, it'll become an arm cell or a bone cell or a muscle cell. And in these guys, they're able to do that throughout their entire lifetime. So they're able to take cells that have been differentiated and determined to be a bone cell or a muscle cell, and they're able to de-differentiate them and put them towards being something they need at that point in time, which is absolutely incredible. Again, that's something that no other species really can do past their embryonic form. There has been research started in these species for the past like 20 years on mice and on some human cells on being able to regrow digits, so your fingertips and such, but typically only is able to occur in very young individuals at this point in time. So there needs to be more research into this because being able to utilize stem cells to regenerate things like appendages is something that could eventually lead us to be able to develop regeneration for other things such as organs. In salamanders, there are some species such as axolotls that are able to regenerate heart tissue. They're able to regenerate brain tissue throughout the whole of their lifetime. So it's very like insightful to be able to learn how to regenerate for the process of utilizing it for medical information. All right, we're going to take a brief break before I kind of continue. We're done talking about anatomy for now. We're going to go into ecology in a little bit. But if there's any questions at this time, feel free. I'm going to stop sharing my screen so you guys can see my face for a second. All right.
Well, thank you, Hadley. You've been doing such a great job. I think everyone can agree with me that it is very interesting and a very awesome presentation that you've been giving so far. Before we get into the Q&A on the floor, I think we can ask some of our own questions. So what really drew you to salamanders? Like, what inspired you to take that path course?
Yeah, so when I was in undergrad, oh, sorry, can you hear me?
Yeah.
Yeah, okay. When I was in undergrad, I took some analytical chemistry courses and was really interested in their the water chemistry side of things, in how complex the systems were in such as like calcium hardness in the water and pH in the water and nitrogen cycle within the water and how that impacted amphibians specifically. Amphibians, because they have that semi-permeable skin that they can breathe through, are super, super influenced by inputs into the water. And being from an area that has a lot of agricultural use, I saw impacts of agricultural use on amphibian diversity within the areas I lived. So being able to see that firsthand kind of drew me to amphibians in general. And I love frogs, I really do, but amphibians like salamanders really have my heart just because of the variety within them and the opportunity that I had to work with a species that was state threatened was something that really pushed me to work with salamanders in the first place.
That's so fascinating. Thank you so much. So is there, in terms of this process of regeneration, is there any sort of limit? Like how many times, like is it only a one-time thing if they just get it cut off the first time? Like what if that regenerated limb, sorry.
Yeah, so they are able to regenerate it multiple times. It does take a while to regenerate though. So if you cut off the arm, it's going to take months to get back to that stage. If you take a chunk out of their brain, it's going to take a moment for them to be able to be back to their full self again. There's still research going on as well on if their memory is retained in brain damaged individuals. So there needs to be more research in the way of regeneration. Impacts, but they are able to regenerate multiple times from the same area, which also kind of goes along with their defense mechanism of them losing their tail when they feel threatened. They're able to regenerate their tail, they're able to regenerate their limbs because they need to be able to for their defenses and for their future survival.
That's interesting. How big do they grow and do different colors mean anything?
Yeah, so there's a large variety of sizes. A lot of individuals tend to stay within like a half foot size. Some stay a lot smaller than that as fully grown adults. So this bright orange guy I'm working with that has those like chin barbells on him, um, he's really is maybe 4 inches fully grown. But there's also some individuals, such as the American species called hellbenders, they can get to be a couple of feet long. And then there's a giant Asiatic salamander that can get to be 5 to 6 feet long. So quite a long range. The Asiatic salamanders are also wide, so they retain the salamander shape, versus the greater sirens that don't have those limbs and look just like a long eel. Those ones can also reach lengths like 4 or 5 feet, but they retain a smaller tubular shape for the water they navigate in the cave systems that they utilize.
Great. So before we head into some of our other questions, I encourage everyone that's watching right now to leave some questions for Hadley in the Q&A portion. But with that being said, our previous presentation was referencing venomous snakes. Are any salamanders poisonous?
Yeah, so there are some that have toxins on their skin, that would nothing to the extent at which you would die necessarily, unless you were to get lacerations or infection from eating something. All amphibians and reptiles and birds have the capacity to have things like E. coli and salmonella on their skin. So they have the capacity to wreak havoc on your system and potentially lead to death. But there isn't any that have true venoms that could inject you and cause any issues for you.
Yes, perfect. Thank you for answering that. Can salamanders be pets?
Yeah. So salamanders definitely can be pets. A lot of people utilize salamanders for outreach individuals, such as mole salamanders, like that tiger salamander I was mentioning. I know it's the Illinois state amphibian. It's a state amphibian for a ton of different states. They're nice, charismatic guys. They've got chunky little faces, and they're very cute and very interactive, I'd say, in that they tend to come out a little bit more readily than some of the other individuals, like these stream salamanders I'm working with. There's also some fully aquatic individuals that people tend to keep as pets. Axolotls, for example, are something that is kept in larger populations in captivity than they are in the wild, which is actually super important for that species survival because currently they only have the capacity to live within one lake. They originally came from an area near Mexico City, or where Mexico City is, which was originally a island that had canal systems built out for farming. And eventually, as the city expanded, a lot of those lakes and canals got kind of soil put into them so the city could expand, and those lakes were drained fully so that the city was able to continue to grow and become one of the major cities in Mexico today. The bad thing about that is there's only one lake remaining in that area that has them, and they're trying to reintroduce them into areas that habitats that are similar to what they need. So the individuals that are keeping them in captivity today are super important and able to introduce genetic variety into that small population so that there isn't any bottlenecking within the population that could cause impact on them.
Great. So just one more question from us. So who are like salamanders closely related to? Would that be geckos and newts?
Yeah, so newts are a type of salamander technically. Um, the closest relation would be frogs, just because they have those similar skin type, similar respiration type, similar heart. So the way that they evolved is a lot more similar to frogs than it is to other lizards. Um, but yeah, I can go ahead and get into the rest of the presentation. Thank you so much for asking me those questions. I will share my screen again. All right. All right, there you guys go. So moving on. Gonna go a little bit into the ecology of these guys. So we know a little bit now about how they work. Now we need to know, why do we care at all? What do they do for the population and the biodiversity of the areas that they live in to be able to help these areas stay viable as an ecosystem? There's two major things that these guys do, and I am so sorry for the quality of this photo. We'll go back to this guy. He looks a lot better. So the main thing that they do in the water systems where they are is they help with the distribution and the diversity of the macroinvertebrates where they live. I know when I was at EHS, in the environmental science class, we did a lot of stream sampling at the stream outside of the school, and we were able to see a little bit of what the species of macroinvertebrates there were, and that's a great indicator of water quality. The more diversity that you have, and the more species that are more like, there's individuals that are able to survive in anything. And there's individuals that are super, super sensitive. So when you have a good mix of those, and you have more of those sensitive individuals that need more pure water or need water that has less inputs into it, then you're able to kind of tell that you have a healthier ecosystem there. Salamanders are able to provide input into that as well, in that, one, they have that semi-permeable skin where they are super dependent on the water quality within the areas that they are, because they absorb water and air directly into their skin. They are able to drink, but they mostly do water absorption and air absorption through their skin. So being one with the water is something that is a great indicator for humans to be able to tell what the quality of that system is, and to make sure that we're creating laws and regulations that protect those water bodies. We call species that are able to tell us a lot about these areas indicator species. So macroinvertebrates and amphibians are definitely within that group, in which they are able to help indicate to us what, if there are issues that are present in the area. They're also super integral in creating a diverse macroinvertebrate selection that isn't too oversaturated with certain individuals. A lot of times you'll find areas that have multiple species of amphibian and salamander that are able to occupy different niches, and they are able to eat different individuals based off of their size, based off of their habits. Different individuals stay within the stream for longer because they're larval for longer, or there's some individuals that stay in their larval stage for up to 4 or 5 years. There's some that stay up into their larval stage forever. There's some that are in their larval stage for only 6 months. So they're able to impact the water diversity within those macroinvertebrates differently depending on how long they're in there. And that allows them to be like a key indicator for the ability for those species to proliferate and also retain the complexity that they need in that area. So there is not too much oversaturation of detritivores, like insects that allow for decay of certain plant matter and organic matter. So they're able to help mitigate those population levels so that things are balanced. They also are able to help with soil health. Soils that are compacted are not very healthy soils because it makes it difficult for things to pass through them. It makes it difficult for insects to go through them and other species to go through them, including mushrooms and plants and trees. So having soils that are well aerated are super important for the ability for these plants and mushrooms and insects to be able to respirate, to be able to have mobility, and to be able to stay within the environment and not create like kind of a dead zone. Salamanders are integral in that a lot of them do have digging capabilities, such as the small salamander. They have like a little bit beefier front arms, and they're able to push the soil back and dig. A lot of salamanders also utilize tree holes, so they'll follow the roots down, or they'll follow fallen tree stump holes down into the soil to be able to help aerate that system and stay at a more consistent temperature when it is winter time or when they're trying to stay out of areas that are drier. So soil health and water health are two really, really important ways in that salamanders help the environment and are integral to being able to provide these niches that this whole ecosystem needs to be able to survive. So why does it matter? And what am I doing about it? So the reason why salamanders matter in general is because biodiversity is important to be able to provide ecosystem health and make sure that these systems are able to retain viability. And I am trying to continue research in these areas so that we have the capacity to retain these species, avoid creating sinks or areas that can cause extinct populations or extirpated populations, so that we have the ability to know more about these individuals and what our inputs into the environment are doing to them. In the project I'm working on right now, one of the things I'm looking at majorly is the impact of pesticides on these individuals. We need to utilize pesticides to help our food production, but also to help these salamanders. They depend on the shaded regions providing cool temperatures to the streams that they live in. And if these insects are able to decimate the tree populations in these areas, they will not be able to survive there either because the temperatures will rise in the streams. So we need to have a good balance of how much risk is there associated with applying a pesticide that is helping keep these trees alive versus what kind of impacts are these pesticides doing to non-target individuals, such as other pollinators like bees or aquatic invertebrates or salamanders. So my project is part of making sure that these ecosystems are able to stay healthy and maintain that biodiversity, and where there isn't detritus buildup because the population of dragonfly nymphs are too high to have detritivores that are able to break down some of those smaller organic matter pieces. So I'm trying to help figure out what a good cost-effective, or not cost-effective, but risk-adverse way to maintain these areas is. I mentioned that I work in histology, and my project involves histology as well. So this is a salamander brain. You can see his eyes at the very top where they have those big purple splotches with the red dots in the middle. Those red dots are the lens, which we all have lenses in our eyes. They're that in that central area where they're a little bit firmer. Um, and you can see that very pale area in the middle. I'm not sure if you guys can actually see my mouse, but it's this light purple area um, where it's long and has this like split area in the middle, and it goes into this more solid area at the very bottom. That is a salamander brain. And that's what I'm looking at to see if there are any morphological changes within the brain when it's applied, or when there is a midichloprid applied to the system. I'm also backing that up with behavioral data that will see if any of these effects that we're seeing in the brain are translated to their behavior and their capacity to carry out their life history. Are they able to reproduce effectively? Are they able to hide and defend themselves from predators effectively? And are they able to acquire prey effectively? So hopefully that makes sense to everybody. And I'm going to go into one more question session so that we can round out this talk. All right.
That sounds great. Thank
Thank
you
you.
so much for your presentation. I think we all are going to give you a great big round of applause for that.
Thank you.
That was wonderful. So let's head into some Q&A. First off, how do salamanders act as bioindicators of environmental health?
Yeah, so salamanders have that semi-permeable skin and are a little bit more readily susceptible to things like temperature changes because of their ectothermic. They're more impacted by pollutants and toxicants in the environment. So they're able to be an indicator of if there are impacts in the environment, they're going to get hit hardest the first before fish get impacted. And before we get impacted down the line, we're able to see within the amphibians that something is not right.
Awesome, thank you. So Jeremy Burnham says, as a Georgia Bulldog, are you considering petitioning the university to change the mascot to the salamanders? And he said, as an alumni, I would support it.
Honestly, that would be pretty cool. I know the Bulldogs have a pretty big thing going on down here, though, so I feel like they might be against it. Go Dogs, though.
Awesome. Be necessary for humans to regenerate like salamanders?
Can you repeat that one?
Yes, what would be necessary for humans? What would be necessary for humans to regenerate like salamanders?
Yeah, so the big thing is we would have to be able to determine how we can stop our wound cells from creating a scar as opposed to creating that wound layer that is able to uh, continue to produce new growth. We also would need to be able to figure out how to have stem cells throughout our lifetime. Whereas when we're embryos, we have stem cells readily available, which is why we're able to regenerate when we are in an embryonic stage. But a lot of the times as we age, as we become young, and as we get older, we reduce the amount of stem cells in our body. So being able to have dedifferentiated cells or stem cells, or being able to utilize stem cells in a way that can be injected or utilized to grow new limbs in vitro and apply it to the human body is something where lots of people are currently doing research on, and we need more. So if you're interested, definitely go to college and get some more information on it.
Yes, thank you. I mean, I definitely think we're pretty far off from figuring that out for now. Next, how do habitat destructions impact salamander biodiversity?
Yeah, so different salamanders have very specific needs. Hellbenders, for example, are in areas that need very clean, clear waters and fast-moving streams. And they need to have areas that have lots of rocks to be able to hide under. And the biodiversity in that area is impacted by their size when they're larvae, when they're larger, and also by the other salamanders that are in that area. different sized individuals can eat different prey items. So individuals that are larger are able to eat macroinvertebrates that are larger, such as dragonfly nymphs and chironomid larvae or midge larvae. That allows them to kind of set up what the impact of detritivores in the area would be. So individuals that eat smaller larvae are able to mitigate what. So sorry. I'm trying to figure out how to say this more chronologically. When we are in fall, we have lots of leaves falling down, and we need detritivores to be able to break those down in the stream surfaces so we don't have an accumulation of organic matter. Or we don't have decay happening that increases nitrogen content or decreases the pH in a certain region. So different sized detritivores, like our different sized insect larvae, are able to break down different parts of these and are prevalent in areas in which they're different sizes at different times of the year. So salamanders that have longer larval stages or at different sizes are able to help mitigate the way that things are broken down within that system. So based off of timeline in which they are in that system. So if an individual is larger and they're in the system for longer, such as those 4-year-old larval black belly salamanders, they're able to eat larger larvae, which increases the amount of smaller insect larvae in the system, and those smaller insect larvae are then able to break down smaller bits of detritus. Sorry for that convoluted answer. I hope
it's okay. Unfortunately, we're nearing the end of our time, but would you be all right with staying a little after to answer the remaining questions?
Of course, yeah.
All right,
Thank you
great.
guys.
Of course.
So they seem to like wet areas. What would happen during seasonal droughts in an area? Is climate change affecting their population?
Yeah, so climate change and droughts are readily affecting salamanders. In areas in which it's becoming drier, you find a lot of reduced abundance of these individuals because they need to breathe through their skin. In areas that are drier, they aren't able to survive. They're able to make it by burrowing underground or finding areas that stay moister. A lot of the times they'll go into tree stumps or tree holes to try and find those areas that are retaining that last little bit of moisture. But if areas become too dry, they aren't able to survive because they can't breathe anymore. And if they can't breathe, they are not able to live. So areas that are experiencing droughts more through climate change and areas especially that are increasing in temperature are impacting the ability for these species to survive. I mentioned the hellbenders and also the stream salamanders I work with require cooler temperatures to be able to work through their bodily mechanics, such as digestion and movement. And as temperatures increase in streams by having climate change occur or by having these trees die off, increasing the amount of sunlight that is able to reach through the canopy, the system starts to break down and those salamanders no longer there and the amount of detritus in the streams increases, which decreases the amount of water in the streams because those leaves and those additional trees are just breaking or sucking up more and more water. So it becomes this cycle that's very difficult to manage, but it can be done. We just need managers that are capable and interested.
Certainly. Thank you. So how does pollution such as pesticides impact salamanders?
Yeah. So different pesticides impact salamanders differently dependent on their mechanism. So there are toxicants that are able to impact the brain, such as the one that I'm working with right now. It's specifically created to target acetylcholine receptors in the brain, which impacts the salamanders, or insects is the target species. It impacts their ability to breed primarily, and also for their liver and their kidneys to function. So for their ability to create hormones and to filter out toxicants in their own bloodstream. other pesticides cause like direct death just because of the way that the toxicants create imbalances within the hormones. Some of them cause more direct impacts on the endocrine system, in which they interact directly with the hormones and cause blocking within the bloodstream, which does not end well and creates extirpation within populations because they're no longer able to reproduce effectively. So, population decline is something that is super prevalent from pesticides, fungicides, and other pollutants that are entering the environment, just varying depending on the concentration that is in the area, which is why laws like the Clean Water Act are super important, and areas like the EPA are super important to be able to create regulations and make risk assessments based off of the needs of the streams in the area that have pollutants entering the system readily.
Great. So just one final question. This one talks about if you could talk more about your fieldwork skills and statistical techniques and or the environmental issues and approach to studying. I feel like this could be like a very elongated answer. I mean,
Yeah.
is it somewhere that like this person may be able to like reach you? Are you on any like social medias? Do you have a page perhaps?
Yeah. I'll leave my email address
Mhm.
is my name. So it's hadley.copeland@gmail.com. I'll go ahead and leave that in the chat really quick if anybody
Yeah.
wants it.
And then you
Um,
can answer the question briefly if you'd like.
and yeah, I'll also answer the question. So, I've gained different analytical techniques throughout my college career and also throughout my work experience. Um, when I was in undergraduate, I took analytical chemistry, and my thesis went over a 2-year period in which I specialized in ion chromatography and flow injection analysis of different chemicals within the water, namely nitrogen and calcium. And then when I started working for the DNR, I also had some more experience with regular water quality parameters such as pH and turbidity. And then we use multimeters typically for that. And we also collected water samples to test for blue-green algae and E. coli. So I learned ELISA for those. I also worked for the state hygienic lab in which we did macroinvertebrate surveys and we did fish surveys through electrofishing to be able to determine species biodiversity within those systems and be able to determine a system health utilizing the conjunction of those macroinvertebrates like you guys utilize at EHS's streams in conjunction with those other analytes like the pH and the nitrates that are in the system. That was specifically for policy. So it was cool to be able to see how legislation and rules were able to kind of impact the way that science was done in the field. I also learned some radio telemetry skills when I've been tracking snakes in the field, as well as trapping techniques with both frogs, salamanders of different varieties, and snakes and rodents as well. Currently, I am in my second semester of my master's degree, and I've taken a statistics course in modeling and being able to utilize different statistical analyses such as ANOVA and t-tests, which are super important and differ dependent on the way that you design your study. Study design is super important to get on the front end. So make sure that you have statisticians that are able to be on your side and people that are well versed on the topic that you're interacting with to be able to make sure that you're providing statistical analyses that are able to make sure that your data is relevant and is able to put something forth into risk assessments and into being able to manage these lands better. So I hope that answered everything. Again, email me if you have any further questions. I'm very thankful for this opportunity. So
Yes, and we want to just emphasize how thankful we are to you for coming again, and we hope to see you next year. Thank you so much from
Thank you.
all of us, and we'll give you another round of applause.
okay, thank you. And great job with MBTI this year, guys.
Thank
Excellent
you.
work.
Have a great rest of your day.
You as well.